Hello everyone, this is Yashwin from Exam Help Lab. In this video, I'll be solving paper 2 variant 1, October November 2023 AS level biology. Starting with question number 1. Scientists measured the concentration of sodium ions and potassium ions in the red blood cells and the blood plasma of a group of people. The results are shown in table 1.1. A part 1 asks you to use the information in table 1.1 to identify and describe the process by which potassium ions enter red blood cells from blood plasma. Okay, so here's the column for potassium ions. As you can see, potassium ions have a higher concentration in red blood cells compared to blood plasma, and the question wants you to identify the process by which they enter red blood cells from the blood plasma. So movement of ions from an area of their lower concentration to an area of their higher concentration occurs through active transport. So here you're supposed to describe the process of active transport, which is against the concentration gradient using ATP. Part 2. Sodium ions and oxygen molecules enter red blood cells. State one similarity and a difference between the processes used by sodium ions and oxygen molecules to enter red blood cells. Let's look at the difference in concentration of sodium ions between red blood cells and plasma. Clearly, there is a high concentration of sodium ions in blood plasma compared to red blood cells. So they're going to move into red blood cells by diffusion, and sodium ions being charged means that they will be repelled by the hydrophobic nature of the cell membrane of red blood cells. So they need a channel protein for the transport, as in sodium ions would move into red blood cells by facilitated diffusion. Oxygen also moves into red blood cells by the process of diffusion, but that's just simple diffusion, since oxygen is a small non-polar molecule, so it can easily diffuse between the hydrophobic layer of fatty acid tails in the cell surface membrane of red blood cells. So the similarity is that both of them move into red blood cells by diffusion, and oxygen, being a non-polar molecule, moves into red blood cells by simple diffusion, whereas sodium ions will need channel proteins. Part 3. Chloride ions move across the membrane of human red blood cells in a process called the chloride shift. Explain why the chloride shift is important in the transport of carbon dioxide from respiring tissues. The chloride shift occurs when carbon dioxide, which is a byproduct of respiration or the waste product, when carbon dioxide diffuses into red blood cells. So carbon dioxide is produced by all of the body cells that carry out respiration, and it's carried away from the tissues and the cells of the body via the blood into the lungs, where it can be exhaled. So while carbon dioxide is being carried by the blood, it will diffuse into red blood cells, where it will combine with water and undergo the reversible reaction to form hydrogen carbonate. Now this reversible reaction is catalyzed by carbonic anhydrase enzyme. Hydrogen carbonate will then dissociate to form hydrogen ions, also known as protons, and bicarbonate ions. These protons will remain in red blood cells, while bicarbonate ions diffuse out of red blood cells via a transport protein. The same transport protein that transports hydrogen bicarbonate ions outside will also move chloride ions into the red blood cells to prevent an electrical imbalance. So in the red blood cells, you will have protons building up because you've got carbon dioxide resulting from respiration, which had produced an acid. The acid will dissociate to produce protons, which cause an acidic pH. Now this acidic pH is balanced. The body has a mechanism to balance this acidic pH. by moving negative ions into the red blood cells. And since the ions are negatively charged, they are polar, that means they'll need a transport protein. Chloride ions will need a transport protein to move into red blood cells. This is the same transport protein that will move bicarbonate ions out of the red blood cells. If chloride ions did not move into red blood cells, there would be an excess of protons in the red blood cells, and this would produce an electrical imbalance. B. Scientists studied the uptake of a substance F by human red blood cells. The red blood cells were immersed in a solution of substance F for 30 minutes. After this time, the scientists recorded two observations. The cell surface membrane of red blood cells showed enfoldings, also known as invaginations, and they also showed an increase in the number of vesicles in the cytoplasm. So it's almost like here you can observe the cell surface membrane before being immersed in the solution, and afterwards you would see something like this, foldings of the cell surface membrane pointing inside the cell. This is an indication of endocytosis, as during the process of endocytosis, vesicles outside of the cells will join with the cell surface membrane and release their contents into the cell. Identify the process by which substance F entered red blood cells. Again, this would be endocytosis. When a substance moves into a cell by forming vesicles, this is endocytosis, and the invaginations are observed when the cell surface membrane folds into the cell to allow endocytosis by forming these vesicles. Question 2. Figure 2.1 is a transmission electron micrograph showing a section of a specialized epithelial cell found in the lining of the stomach. This cell produces extracellular proteins that are released into the bloodstream. So transmission electron micrograph is shown in gray color as usual. Part A asks you to outline the role of structure X and structure Y as shown in figure 2.1 in the production of extracellular proteins. Epithelial cells are cells that are found lining many body parts, for example, the skin and blood vessels. And in this case, these epithelial cells are found lining the stomach. To recognize structures X and Y, let me switch colors to blue. So to recognize X and Y, you would need to identify this entire membrane bound organelle as the nucleus, which is surrounded by these dark spots, which are ribosomes, and these structures as vesicles. So now that we have identified the larger organelle as the nucleus, we know that nucleus has got chromosomes in the form of chromatin, and it's also got a nucleolus. So X seems to be a concentrated mass, so that would make it the nucleolus, which is the site of ribosome synthesis, and Y is a large amount of thread-like structure inside the nucleus. So it's going to be the chromatin. Chromatin essentially contains the genes that code for polypeptides. Part B, the cell in figure 2.1 releases ghrelin, which is a small protein that acts as a cell signaling molecule. So cell signaling molecules essentially bind to the cell surface membrane of a cell, and they can speed up or slow down the rate of reactions that occur within a cell. Figure 2.2 shows the sequence of amino acids in a ghrelin molecule. So essentially the primary structure, a sequence of amino acids, the primary structure, The amino acid serine is in position 3 in figure 2.2. So here you've got the position they're talking about. Figure 2.2 has been modified by the addition of a saturated fatty acid chain. So a saturated fatty acid chain is one which does not have any carbon-carbon double bond, and all of the carbon-carbon bonds are single bonds. Part 1 asks you to state the level of protein structure shown in figure 2.2. This is a primary structure since you only see peptide bonds in the polypeptide and there aren't any hydrogen bonds which would have produced a secondary structure, or ionic bonds, disulfide bonds, and hydrophobic interactions that could have also made up a tertiary structure. Part 2 state one similarity between the structure of a saturated fatty acid molecule and an amino acid molecule. So an amino acid molecule essentially consists of a carboxylic acid group as well as an amine group, and then the central carbon atom is bonded to a hydrogen atom as well as an R group, which the simplest form is a hydrogen atom. This could also be an alkyl group, for example CH3, and a methyl group, and so on. So this is the basic amino acid structure. Now you're supposed to spot the similarity between this structure and a saturated fatty acid molecule. Well, both of them would have the carboxylic acid group, so that's your similarity. This one over here is propanoic acid, which is a saturated fatty acid. So here you can also see the carboxylic acid group. Part 3, the addition of the fatty acid chain allows ghrelin to function as a cell signaling molecule. So just how the addition of this fatty acid chain allows a ghrelin molecule to act as a cell signaling molecule. So here you need to understand the mechanism by which a cell signaling molecule works. So as I mentioned earlier briefly, these molecules are supposed to bind to the cell surface membrane of the cell that they are going to target, and for that to happen they need to have a shape that's complementary to the receptors. So let's say this is the target cell. It's going to have a receptor, and this receptor will bind to the cell signaling molecule. Let's say this is the ghrelin molecule alone on its own. It doesn't have the saturated fatty acid, and as you can see, this cannot bind to the this fatty acid alone cannot bind to the receptor site over here. Now if you were to add the fatty acid molecule like so, this shape would be complementary to the receptor site, allowing the cell signaling molecule to bind to the cell. So a cell signaling molecule can only bind to the target cell when it has a complementary shape to its specific receptor. The fatty acid over here will modify ghrelin's three-dimensional structure by giving it a shape that's complementary to its receptor. This will allow ghrelin to bind to the receptor. Part C, scientists have discovered that the gene coding for the ghrelin contains 5,000 base pairs. So these base pairs are the pairs of nitrogenous bases found on the two strands of the DNA. So that's one base pair, and you're going to have 5,000 of these in the gene that codes for ghrelin. This is a much larger number of base pairs than is needed to code for the ghrelin molecule shown in figure 2.2. Part 1 asks you to calculate the percentage of base pairs found in a gene that codes for the ghrelin molecule in figure 2.2. You're supposed to show your working and give your answer to one decimal place. You'll need to count the base pairs in figure 2.2, which comes out to 28 amino acids. Now, each amino acid is coded by 3 base or a triplet. and this would need 3 base pairs, which means that 28 amino acids would need 84 base pairs. Other than that, you'd also need a codon, a start codon as well as end codon. So that would be an additional 6 base pairs, 3 for starting and 3 to end. And they want you to calculate the percentage of base pairs in the gene that code for the molecule. Alright, so you're supposed to add up the genes that are used, divide them by the total number of, you're supposed to add up the base pairs that are used, divide by the total number of base pairs and multiply by 100 to find the percentage of genes that are used. So that's going to be 28 multiplied by 3 plus the codons for starting and ending divide by 5000 multiplied by 100, and this should come out to 1.8%. Part 2. The primary transcript produced from ghrelin gene is a longer molecule than the mRNA found in the cytoplasm. So the primary transcript is the first product that you gain from translating the DNA. Explain how the primary transcript is modified before translation. So RNA transcription is split into 2 steps, and RNA transcription will produce the mRNA that they are talking about over here. The 2 steps are, let me just erase this. So the first step is pre-RNA production. In this first step, the pre-RNA that's produced will have non-coding parts, also known as introns, as well as the coding parts, known as exons. The second part of mRNA transcription is the modification, which will produce mRNA. So modification, this part will remove the introns or the non-coding parts by splicing them out and joining the exons together to produce mRNA. This process is called RNA splicing. So as a result of first step, you will have exons which will code for the protein, bind it to, let's say this is here's some intron bases that are the non-coding part. Let's say this is the result of the first step, which was production of pre-RNA, and then the introns or the non-coding parts will be cut out to produce the result, which will contain only the exon. This is the result of the second step, or RNA splicing. Now this is why the primary transcript, this over here, which is produced from the gene, is much longer than the mRNA molecule. And this step is necessary so that it becomes easier for the smaller molecule to move out of the nucleus towards the cytoplasm, where the mRNA will be used for translation. Question 3. Plants have specialized cells for the efficient transport of assimilates. Assimilates are the organic compounds found in phloem tissue, that is sucrose mainly. Table 3.1 shows some of the features of 2 different types of cell found in plant tissue which are adapted for the efficient transport of assimilates. Cell type A and B are found in phloem tissue. Many plasmodesmata connect type A cells with type B cells. Identify cell type A and explain why plasmodesmata are important. So the two types of cells found in phloem sieve tube are sieve tube elements and companion cells. Sieve tube elements are where the main flow of assimilate occurs, or this is where the organic compounds will flow through, whereas companion cells have two functions. They control the metabolism of sieve tube element, and they also aid while loading and unloading of sugars into the phloem. Let's look at cell type A. It's got cytoplasm and cellulose in the cell wall. It does not have nucleus and ribosomes, which indicates that it doesn't produce proteins on its own, and it's got only very few mitochondria. The absence of nucleus and ribosomes and the very few mitochondria will reduce the friction to the flow in phloem sieve tube element. Therefore, cell type A is phloem sieve tube element. And as to why the plasmodesmata are important, let's say you've got two plant cells connected together, and you want water to get transported between these cells. There are two ways that water can pass through them. Either it would diffuse through the cell walls, which is the apoplast pathway, or water could simply diffuse through the gaps in cell walls. Also, these gaps are plasmodesmata. This is the symplast pathway, which is much faster compared to apoplast pathway. So plasmodesmata is the area in plant cell body without cell wall. It connects the cytoplasm of companion cells and sieve tube element, which would provide symplast pathway for sucrose to diffuse into sieve tube elements. So symplast pathway increases the transport efficiency between plant cells. If sucrose had to diffuse through the apoplast pathway or through the cell walls, that movement by diffusion through cell wall would take much longer, so it would have reduced the efficiency. Part B, the polysaccharide callose is found in the cell walls of cells close to plasmodesmata. Figure 3.1 is a simplified diagram showing the structure of callose. Not all hydrogen atoms are shown. Complete table 3.2 to compare the structure of a callose molecule with a cellulose molecule. Alright, so you need to be familiar with the structure of cellulose molecule to answer this question. First, you're supposed to compare the monosaccharide used to synthesize the polysaccharide. So callose molecule as given in the question is produced by the beta glucose monomers, and cellulose molecules are also composed of the monomer beta glucose molecules. And here's a beta glucose molecule for comparison. Now you're supposed to compare the bond connecting the monomers in a callose molecule and a cellulose molecule. In a cellulose molecule, the beta glucose molecules are connected by 1,4 glycosidic bonds. So this is carbon atom 1, 2, 3 and 4. Now the bond connecting monomers in a callose molecules are 1 and 3 glycosidic bond, as the bond connects the first carbon atom and the third carbon atom in adjacent monomer molecules, and it's a glycosidic bond because you've got carbon, oxygen and carbon all single bonded. Now the shape of a callose molecule is a helix, and you're supposed to describe the shape of a cellulose molecule. Well, a cellulose molecule has a straight or linear shape. And as for the orientation of monosaccharides in the molecule, in cellulose, the alternate glucose molecules are rotated by 180 degrees to allow the formation of 1,4 glycosidic bonds, whereas in the callose molecules, all of them have the same orientation. As you can see, all of the monomer molecules have the CH2OH group on the top, which shows that all of them have the same orientation. Part C. Hydrogen bonding is important for the movement of water through xylem vessels in a plant. Describe the roles of hydrogen bonding in the movement of water through xylem vessels. Here you need to know the role of hydrogen bonding in transpiration, as it's because of the loss of water vapor through transpiration that causes water to move up the xylem vessels. So as water vapor is evaporated from the surface of spongy mesophyll cell walls, water is drawn up in the xylem vessel by transpiration pull. Water moves from the top of xylem vessels towards the mesophyll cells after evaporation of water vapor. And that's because between water molecules, hydrogen bonding will exist, which will create this water column. So hydrogen bonds between water molecules will draw water molecules towards the surface of the xylem. Hydrogen bonding is the reason for the adhesion force which occurs between water molecules and cellulose in the xylem wall, so that water molecules are attracted to these molecules. And hydrogen bonding will also cause the cohesion force, which is a force between water molecules. This forms a continuous column of water through xylem. Water vapor will be evaporated from the cell wall of spongy mesophyll cells, from the surface of spongy mesophyll cells, and due to attraction between water molecules resulting from a hydrogen bonding, water molecules at the top of the xylem vessel will be drawn towards the spongy mesophyll cells, and these molecules at the top will then attract the water molecules lower down, and this is how the transpiration pull will continue to pull up water. Now, part D. Water that has traveled through xylem vessels reaches the leaves. Cooling of the leaf occurs as a result of the evaporation of water during transpiration. Water has a high latent heat of vaporization because water molecules form hydrogen bonds. Well, during vaporization, what happens is bonds between water molecules will break, bonds between liquid water molecules will break down to produce water vapor, and this will require energy. And because these bonds are hydrogen bonds, meaning strong bonds, this will require a lot of energy to break bonds between liquid water molecules to form water vapor during transpiration. With reference to hydrogen bonding suggests why cooling of the leaf occurs as a result of evaporation of water during transpiration. So during evaporation, hydrogen bonds between water molecules are broken down, and this will use up heat energy from plant. So breaking bonds will require energy, and this is provided by the heat energy from the plant body. As heat energy is used up, cooling effect is caused. Question 4. Some people who are infected with HIV develop AIDS. In part A, figure 4.1 shows the number of people that have been newly infected with HIV. So you're shown new infections in 2018 across the world and the percentage changes in the number of new infections since 2018. So they're showing you the current number in 2018 and the changes since 2010. All right. So for example, A shows that there are 68,000 new infections in 2018, which is a 12% decrease from 2010. So in 2010, there was 12% more than 68,000 infections of HIV. Part 1 asks you to state the full name of the pathogen HIV, which is the human immunodeficiency virus. Part 2, using only the data in figure 4.1, state what can be concluded about the change in number of new HIV infections across the world between 2010 and 2018. You may use the letters in figure 4.1 to identify the regions of the world. One thing before proceeding, I'd like to point out is that HIV is the human immunodeficiency virus, which is the cause, and AIDS is essentially the acquired immunodeficiency syndrome, which means the symptoms that someone is infected with HIV. To answer part 2, you need to pay attention to the word state, meaning that you need to quote and describe the change shown by data in figure 4.1. Do not include any explanation or the potential cause of the change in your opinion for the answer. You're just supposed to state the data shown in the figure. This is a 4-mark question. I would suggest you to start with the general trend and then also mention any exceptions to this trend. So generally speaking, the number of HIV infections decreased in all areas from 2010 to 2018, except for the Middle East and North Africa, as well as the East, Europe and Central Asia region. Let's look over here. So here you can see you've got a decrease, again a decrease, and most of these are decrease in cases, except for 2 regions which are an increase in cases. Those would be Eastern Europe and Central Asia, as well as Middle East and North Africa. So these are the exceptions. The general trend is a decrease in the number of cases. Now you can write down the highest and the lowest values of increase and decrease in the general trend and in the exception. So the greatest increase was the 29% increase in Eastern Europe and Central Asia, and the biggest decrease in the percentage of infections was this 28%. Okay, I probably shouldn't use this green color. So the biggest decrease was in East and South Africa. So in the first part of my answer, I have mentioned the general trend and the rule and the exception or exceptions to the trend, which will allow you to score 2 marks out of the 4. And then to complete your answer, you can elaborate on both of these points by using data from figure 4.1. B. People who develop HIV/AIDS have fewer T helper cells as the pathogen destroys these cells. This makes them more susceptible to tuberculosis. Explain why people with HIV/AIDS are more likely to develop tuberculosis. So TB or tuberculosis is an infectious disease, and HIV weakens a person's immune system by reducing the number of helper T lymphocytes. Now, the function of helper T lymphocytes is to release cytokines. Cytokines will stimulate B lymphocytes to produce antibodies. Cytokines would stimulate B lymphocytes to produce antibody secreting plasma cells, and cytokines will also stimulate macrophages to increase the rate of phagocytosis. Now you can imagine why the
decrease in number of helper T cells would weaken immune system, as not only would the affected person have fewer antibodies, the rate of phagocytosis carried by macrophages would also reduce, which means that the chances of survival of a pathogen inside the body of this person would increase, and therefore this person would be more vulnerable to infectious diseases that are caused by pathogens. So TB would weaken, TB is an infectious disease. HIV would weaken a person's immune system by reducing the number of helper T lymphocytes. Consequently, fewer macrophages carry out less phagocytosis of pathogens, and also fewer B lymphocytes are stimulated to produce antibodies through plasma cells. Both of these events reduce the person's ability to fight off the pathogens and increase chances of infection. Now, this is true for not just TB but any infectious disease, and this increased vulnerability of a person to infectious diseases is what leads to AIDS. A person infected with HIV may develop a heart condition called cardiac tamponade, where the pericardium is a thin sac surrounding the heart, as shown in the healthy heart in figure 4.2. So here you can see the pericardium sac. When a person develops cardiac tamponade, the pericardium starts to fill with blood, which you can also see over here. This prevents the ventricles functioning efficiently. People with cardiac tamponade experience a variety of symptoms, including low blood pressure and an increased breathing rate. Suggest why a person with cardiac tamponade would experience symptoms of low blood pressure and an increased breathing rate. So this is a 4-mark answer. So a person with cardiac tamponade has a pericardium sac that is filled with blood, and as you can see, this will exert pressure from the pericardium towards the ventricles. And the potential reasons for this pressure in pericardium decreasing the blood pressure and increasing breathing rate could be that when blood is filled in pericardium, it would exert pressure on the ventricles. This would reduce the volume of ventricles, and the ventricles would not get completely filled up when blood flows into the ventricles through the atrium before systole. So when the ventricles do not get completely filled up, a smaller volume of blood is pumped out of the heart during the systole. So the ventricles can only pump out as much blood which gets in there. If you don't have enough blood in the ventricles to begin with, they cannot pump out enough blood through systole, and this would happen when the pressure from pericardium would reduce the volume of ventricles, and this would result in low blood pressure, since the volume of blood that's pumped out into the arteries is not the normal amount. As a lower volume of blood is pumped into the body cells would not reduce a sufficient amount of oxygen from the blood. The left ventricle is responsible for pumping oxygenated blood to the entire body, and this oxygenated blood will then supply oxygen to all parts of the body. Now, if the body doesn't get enough oxygen through the oxygenated blood, to compensate for this, the breathing rate of lungs would increase to supply more oxygen from the lungs. In this question, the commanding term was suggest. So clearly they don't expect you to have prior knowledge of this disease. However, you're supposed to suggest an answer by looking at the information given in the question and using using knowledge that you have already learned about the heart, about the ventricles, about their function, and how the function of heart could also determine the function of lungs, how the volume of blood pumped out of the heart could also determine the breathing rate. Question 5. Figure 5.1 is a photomicrograph of a transverse section of a bronchus in the lung. Part A1. Identify the tissue labeled T. So T has got this glassy or amorphous appearance, which you'll be able to see when you zoom in or look closely at T. You can almost see these tiny bubble like structures which identify T as the cartilage tissue. Part 2. Describe the function of smooth muscle in the bronchus. Smooth muscles are found in walls of tracheae, bronchi, and bronchioles. They help to regulate the flow of air into lungs by relaxing when more air is needed. This will increase the diameter, and smooth muscles will contract when less air is needed to decrease the diameter. Part B. The walls of alveoli contain some specialized epithelial cells called type 2 epithelial cells. These cells secrete surfactant. Surfactant helps to prevent the alveoli collapsing during breathing. Surfactant contains phospholipid, cholesterol, and protein. The components of surfactant are synthesized in rough endoplasmic reticulum and smooth endoplasmic reticulum and then passed to the Golgi body. The surfactant that is produced is stored in secretory organelles called lamellar bodies. The surfactant in the lamellar bodies is released onto the surface of the alveolar epithelium by exocytosis, as shown in figure 5.2. Part 1. Each lamellar body is surrounded by a single membrane. Draw a diagram to show the arrangement of phospholipid molecules in the membrane surrounding the lamellar bodies. Here you need to draw a phospholipid bilayer just like you would for a cell surface membrane, showing fatty acid tails and the phosphate head. Here is a rough sketch of the phospholipid bilayer. Part 2. Scientists studying the production and secretion of lung surfactant have discovered that a reduction in cholesterol in the cell surface membrane of type 2 epithelial cells reduces the secretion of surfactant. Suggests why secretion of surfactant is affected by a reduction in cholesterol in the cell surface membrane of type 2 epithelial cells. Here you need to know cholesterol's role in cell surface membrane of any cell. So cholesterol regulates the fluidity of membrane. That is, if there is more cholesterol, the fluid is going to be more fluid, and vesicles can fuse more easily with it. The exocytosis vesicles or the secretory vesicles or the lamellar bodies in this case, and vice versa. So if there is fewer cholesterol molecules in the cell surface membrane, exocytosis won't be that easy. Secretion by exocytosis involves the fusion of vesicles or lamellar bodies, in this case with the cell surface membrane. The ability of lamellar bodies to fuse with cell surface membrane of epithelial cells depends on the fluidity of membrane, which in turn depends on the amount of cholesterol molecules in the membrane. Part 3. Lung surfactant is engulfed by macrophages that are in close contact with type 2 epithelial cells. Suggest why macrophages Suggest why macrophages engulf surfactant. So again, here you're supposed to suggest an answer. You're not expected to know the content. You're supposed to suggest based on the information provided in the question and using your knowledge. So macrophages are immune cells that engulf pathogens. To understand how surfactant could be useful to macrophages, I'd look at its composition. The question tells us that surfactant is composed of proteins, cholesterol, and phospholipid. Now you need to think of how these three substances could be useful to macrophages. One reason for macrophages to engulf surfactant could be to utilize the protein, phospholipid, or cholesterol found in them. Question 6a, part 1, state the name of the phase of mitotic cell cycle during which DNA replication occurs. That phase is the S phase, which stands for semi-conservative DNA replication. Part 2, during research, scientists can use modified nucleotides to prevent elongation of polynucleotide chain during DNA replication. Figure 6.1 shows the structure of a DNA nucleotide found in the nucleus of a cell. So you've got the three parts over there in the nucleotide. Figure 6.2 shows the structure of a modified nucleotide. Suggest how the structure of the modified nucleotide prevents DNA polymerase joining it to another molecule. Let's start with spotting the difference between figure 6.1 and 6.2. In figure 6.2, the hydroxyl group on the third carbon atom of deoxyribose sugar is replaced by a hydrogen atom. So this hydroxyl group is replaced by hydrogen atom in the modified nucleotide. This will change the shape of the nucleotide, and the resulting molecule would not be complementary to the active site of DNA polymerase, which is responsible for elongating the chain. B. RNA aptamers. Aptamers are short single-stranded RNA molecules that can be used to study some infectious diseases. Scientists studying an infectious disease in animals investigated the effect of RNA aptamers on the activity of RNA polymerase that is produced by the pathogen. The aptamers bind to a specific region on the RNA polymerase. So already you're looking at a molecule that binds to a specific region of an enzyme. This should get you thinking about the different kinds of molecules that can bind to an enzyme, which is a substrate or an inhibitor. Table 6.1 shows the effect of two aptamers, F47 and F52, on the activity of RNA polymerase produced by the pathogen. Part 1, with reference to table 6.1, compare the effect of aptamers on the affinity of RNA polymerase for its substrate. The table compares the Vmax, or the maximum velocity of RNA polymerase, and the Michaelis-Menten constant values of the RNA polymerase enzyme when without aptamers in the presence of F47 and F52 aptamers. Before starting, I'd like to explain the meaning of maximum velocity and Michaelis-Menten constant value. So Vmax is the maximum rate at which an enzyme works, and the Michaelis-Menten constant is the substrate concentration at which half of the maximum velocity of enzyme is achieved, or at which half of the active sites are occupied. The greater the affinity of an enzyme for its substrate, the lower the Michaelis-Menten constant value will be. As for a stronger affinity, you won't need as many substrate to fill half of the active sites. Even for a small concentration of substrate, there would be enough attraction between the substrate molecules and the enzymes to occupy half of them, whereas for a larger Michaelis-Menten constant would mean that the affinity of the enzyme for the substrate is smaller, as you would need a greater amount of substrate molecules to occupy half of the active sites. Anyway, so the table here shows an increase in the Michaelis-Menten constant value of enzyme when aptamers bound to it from 346 to 508 and 523, and this indicates that the aptamers will reduce the enzyme activity by acting as inhibitors. Again, affinity here means attraction of an enzyme for its substrate. As the affinity decreases and Michaelis-Menten constant value of enzyme increases, a higher substrate concentration is required to reach the maximum velocity and fill half of the active sites. A higher substrate concentration is required to reach half of the maximum velocity and to fill half of the active sites. So to answer part 1, compare the effect of aptamers on the affinity of RNA polymerase for its substrate. Both aptamers reduce the affinity by, as you can see from the increase in the Km values. The aptamer F52 reduces the affinity more by causing a greater increase in this value. So F52 causes an increase in 346 to 523, which is a greater increase from 346 to 508. Part 2, with reference to table 6.1, suggests explanations for the effect of presence of an aptamer on the rate of transcription catalyzed by the RNA polymerase. As I mentioned earlier, a possible reason is that aptamers are inhibitors. They could act as enzyme inhibitors by binding to the catalytic site or by binding to the allosteric site. If they bind to the catalytic site, aptamers prevent the substrate or the RNA nucleotide from binding to the active site. Whereas if they bind it to the allosteric site of the enzyme, the aptamers would change the tertiary structure of enzyme and therefore its active site's shape. As a result of change in active site of the enzyme, aptamer prevents RNA polymerase from forming a bond with RNA nucleotides or the substrate to form the sugar phosphate backbone. And this is it. We are done with this paper.